Uh, Good afternoon, everyone. Um, my name is Harender, and I am a PMR fellow from Department of Biological Sciences and Bioengineering at IIT Kanpur. So, uh, this will be the continuation of last session where we were trying to solve various questions related to the concepts that we had learned till now. So, this is chapter 9 of class 12 biology, biotechnology, principles and process. So, as earlier continued with the SBS session, so there will be around like 10 12 questions left. And after this completion of these questions, we'll be moving on to the part 2 of the chapter, which will be studying subsequently. So, name the regions A, B, and C. So, this is the plasmid that we were learning, PBR, T22, where there was double selection, I hope you remember, ampicillin and tetracycline. And we want to, like, so in this question, we need to label that, right? Uh, so this is a vector, one. This is a vector, a cloning vector. So let's go. So we know that there are two types of resistance. One is tetracycline, which is already marked here, and C. They are asking what is C. C must be the ampicillin. It is ampicillin resistant. Second, B. B and A are the resistant enzymes. This question is basically asking you to name the enzymes. So I hope you remember that we had one BAM-H1 site, BAM-H1 residual site, and then B we had PSQ1 site, PSQ1 sites. So these are basically nucleases or restriction enzymes or restriction enzymes. Another question. The next question is: a gene was being ligated. So the first word is ligated to a plasmid vector to prepare a recombinant DNA during bacterial transformation. So they're telling us that a gene is being ligated with a vector for the transformation in bacteria, back in a host basically. Right. So here the transformation. So we already know what is transformation. Transformation is a process by which the rDNA is being sent inside the host. Right. Now, so this is what they were doing. Next, an exonuclease was added to the tube accidentally. How will it affect the next step of the experiment? So we know that in the digestion process, we always use endonucleases, which for example, we already know the examples, most common examples like EcoR1, BAMH1, and we recently studying that one like previous like PS1 and something like a lot more. But instead of endonuclease, the one that cuts in between, they added exonucleases. What is exonucleases? Exonucleases are the enzymes that cleave at the end, that cleave at the ends of the DNA strand, right? Now they are saying that the gene of being ligate to a plasmid vector and was vector to prepare and recombinant DNA during bacterial process transformation. But instead of endonuclease, an exonucleases was added to the tube accidentally. How will it affect the next step? So they have added an exonuclease, and now they are asking how will it affect. So basically, there will be no effect on the experiment because because the exonuclease, one of the property of exonuclease is that it cuts at the free ends of DNA. That is at 5' end and 3' end. And we know that a plasmid is completely circular. A plasmid is completely circular. That means there is no free ends. And therefore, the exonuclease, since there is no free ends, there is no 5' or 3' end, the exonucleus will not cut. Will not cut. So remember this. So the entire process was to do normalization and the transformation, but in the first step itself, instead of endonucleases, they added exonuclease. And we know that exonuclease is an enzyme or restriction enzyme that cuts at the end terminals of the DNA. That is at the free end terminals. Free terminals. Basically at the 5' and 3 terminal. But since the plasmid does not have any kind of end, it won't cut. It won't cut. And therefore there won't be any ligation. Moving on, what would happen? What would happen if the restriction enzyme do not cut the DNA specific recognition site? So we know the one property of restriction enzyme making it a robust enzyme is that it cuts only at specific site or a specific region. So we know this, but they are already it's a hypothetical question that what if it does not cut the DNA at the specific recognition site? So we all know that if it cuts randomly or if it does not cut at the specific site, the DNA fragment will be irregular without any sticky ends, and therefore there won't no RDNA would be formed. Because if it is not cutting at a specific site in a specific manner, like if it does not create a sticky end or something like that, that means it won't be helpful in making the RDNA. That is the recombinant DNA. Another question. The question is, what would happen? What would happen if a plasmid without a selectable marker was chosen as a cloning vector? So I hope you remember the selection marker. Selection marker is any gene, any gene that helps to differentiate transformants, transformants from non-transformants, non-transformants. So that was the role of the selectable marker. And the most common example that we did learn was about antibiotic resistance genes, such as AMP resistance, tetra resistance, tetracycline resistance, and cannabis resistance. So this was the role of the selectable marker. But the question is, what if we choose a vector, for example, a vector which does not have a cloning vector, does not have a selectable marker. So I hope you remember that among the four properties and features of a vector, selectable marker was one of the most important one. So if there is no selection marker, so if you are getting a lot of colonies, but you would have a difficult time or difficult situation facing on screening the multiple, like for example, you have 10,000 colonies, right? So in these colonies, you will have from 200, say 200 non-transformants, we do not have your DNA, right? 700, so 800, 800 plus positive DNA. You should have the DNA. But you have to screen each of the colonies to confirm that. So it will be very difficult without a selection marker to select a better transformant. Moving on. Another question here is, what are the properties of a good vector? So again, these are basically features that we're talking about features of a good vector. So what and all a good vector should possess? So we know that it should have OR, right? It should have origin of replication. It should have origin of replication. Then again, in the previous slide, we learned the importance of selectable markers. So it should have a selectable marker, like ORI and selectable marker. Selectable marker. The third very important factor is that it should have unique restriction enzyme sites, right? So that we can easily use it to cut our DNA at a specific size and a specific length. So these three are the most important. And the fourth one, it should be like mostly you will come to know that it should have high copy number. It should be copy number. It should be small and easy to isolate and purify. So these are the common main properties for choosing or being a good vector. Now, what is the source of EcoR1? We know that E stands for genus, Escherichia is there, Escherichia, and CO is for coli species name, R is the strain name, and 1 is the order in which the enzyme was obtained. So this is the first one, right? Mention any three vectorless methods that are used to introduce a recombinant DNA into a competent host. So they're asking about any three vectorless methods. Now that is a bit tricky, right? So we need to have three vectorless method that can be used to introduce recombinant DNA into a competent cell. So till now we were learning about cloning in a vector at that time. giving it to the host, right? Basically transformation, right? But they are having any vectorless method. So I hope you remember the first method that we learned was in the in the last class was the micro injection. So in micro injection, what happens is the our DNA is directly injected into the nucleus of the cell. The RNA is directly injected into the nucleus of the cells because there is no vector. You just take up the DNA, we take up the DNA, and you just put it in the micro syringe, and put it into a cell, into a cell. Second example was ballistic or gene gun. In this, what happens is they coat DNA on gold or gold or tungsten metal balls basically and bombard live cells with it. And the third, so this was first microinjection, second was ballistic gun, third was using bacteriophage. So in this method, we basically pack the DNA into a bacteriophage and then basically deliver it. Next question: which of the following is a palindromic sequence? So I hope you remember what is the meaning of palindromic sequence. Palindromic sequence, I hope palindromic sequence. So there was this example that I gave you back in the class was Malayalam, Mal-ya-la. So this palindromic sequences are sequences which, when read from both ends, pronounce and sound the same. That is, if I write Malayalam from this side, it's Malyam. It's Malayalam. But what if I read from this side? It's again Malyam, Malayalam. Right? So these are in the both sides. If I read from both the sides, that is, if I read from 5 prime to 3 prime or 3 prime to 5 prime, it will be same. Obviously, let's say if I read from 5 prime to 3 prime, it will be same. So there is a 5 prime to 3 prime, it is CGT. If I read from here, 5 prime again, C, again it's not there. So this one is wrong. Next, I have CGA. Here it is C, again it's wrong. Then again, we have GAA. It is in there GAA, TTC, TTC. So this is the right answer that we have because whenever I read this sequence on the prime high prime T, it will always give me the same result. So the chances of mutation or any such thing is a variation in my data. And again, this is the wrong answer. Right? So that's how the palindromic sequences are. And this palindromic sequences, the examples are the restriction enzymes. Restriction enzymes have a palindromic sequence. Which of the following enzyme catalyzes the removal of nucleotides from the ends of the DNA? So they're asking about enzyme, and the second thing they are asking is removal of nucleotides from the ends of the DNA. So this is a very straightforward answer. We know that endonucleus is cut in the middle, and of the answer exonucleus, they cut in the at the ends of the three ends of the DNA. Then you have ligase, it basically joins the DNA, you cannot cut it. And hen 2 is a restriction enzyme. Another question: the transfer of genetic material from one bacterium to another with mediation of a viral vector is termed as. So, what is how is the process? They are asking the process. The transfer of genetic material from one bacterium to another with the mediation of a viral vector is called termed as. So that way it is termed as transcription. Sorry, I already given me the answer. But yeah, transcription is a process by which the DNA material from a DNA material is transferred to a cell by the virus. Next is conjugation. Conjugation is again a type of genetic transfer wherein the bacteria they connect in each other with the pilus, with the opening basically a tube-like opening with them like this, and they will basically transfer this genetic material as well as they create a pilus kind of thing, and they are easily able to allow the DNA transfer. This process is called conjugation. Conjugation. Then we already know what is transformation. We put DNA with the cells and then heat shock 40 degree and transform. And translation is basically machinery where mRNA is translated to protein. So this will be the last question. And after this, we'll be moving on to the part 2. So the question is, how are the transformants selected from non-transformants? So the question is, how is the transformants selected from non-transformants? So how the transformants selected from non-transformant? First is presence of more than one recognition sites in the vector DNA. Okay. Second, presence of alien DNA into a vector DNA resulting in the insertion activity of the signal marker. Okay. Now, antibiotic resistant genes gets inactivated due to the insertion of their DNA. Both is both B2 and 3. Right. Now they're asking us how are transformants selected from the non-transformant. So I hope you remember where we used vector PBR322 where we had this second gene and ampersand gene. In order to first it's a transformation. Uh, First, we selected after putting into tetracycline, we have ampersand plate and we got a colonies. And this from this we were able to differentiate between both the we were able to differentiate between both the transformants as well as the non-transformants, right? So this is the first step uh, of it. Then the second step is uh, uh, first one was presence of more than one recognition site in the vector DNA. So this cannot be the option because the question is asking us to differentiate between the bacteria that has taken up the DNA and the bacteria that does not have the DNA, right? So we have to exclude the uh, first topic basically. Then the second option is presence of alien DNA into a vector DNA results into insertion inactivation of separable marker. Second option is basically presence of alien DNA into presence of alien DNA into the vector DNA results into insertion of inactivation of cell-to-cell marker. So we had cloned the alien DNA into a cell-to-cell marker, right? This one in the tetracycline one. And the third option is antibiotic resistance gene gets inactivated to this. So yeah, so 2 and 3 both are right. So basically, that option 4 is right because we inserted here and then we selected the ampersand. That's how we were able to differentiate transformant from the non-transformant. So I hope you have your concepts clear. And moving on from this, we'll be moving on to the part 2, where we will be understanding the biotechnology, the principles and processes of the part 2 of this chapter. Till now, we have completed what is biotechnology. We have completed what is genetic engineering, the principles of biotechnology. We have, and then we have also completed the basic how the RDNA technology occurs. What are the major steps? What are the tools of RDNA technology? Now we will be applying all these tools and all these principles and understand what is the daily like applications of this. And that's what we want at the end of the day, right? We want how these tools as well as this technology is being used to create uh, to create a to create their RDA technology for for our application. So now the principal part is over. Now the principles part it is over. Now we'll be focusing on the process. What are the process of the biotechnology? The process of recombinant biotechnology. First is isolation of genetic material. Right. First is the isolation of genetic material. So we were like when we want to isolate a gene, right? Like for example, I was giving an example of insulin gene, right? Insulin gene. So this gene, from where do I need a backbone? Like I need to isolate this gene from somewhere. So where is this gene coming from? So where is this gene coming from? We all know that whatever information, whatever our body is coding, whatever our body is giving out, the sequences, the gene, everything, the information, everything is present in the nucleic acid that is DNA for us, right? So this DNA has the gene that codes for this insulin. So if I need to isolate or basically want this insulin that is a protein, I need to go back to the DNA level and check out the gene. Where is that gene? And from there, I can take out the gene and use it to make a product that will be very important for the human use. So first is to get the gene, we need the DNA, right? So first step of the process of RNA technology is the isolation of the genetic material, that is isolation of the DNA. Then we'll cut this DNA, like we want to take out this gene and then incorporate it into our desired plasmid. For that, we need to cut both the DNA as well as the plasmid at a specific site, and then they join them, right? So the second process of the RNA technology is cutting of DNA at a specific location. Third, we can basically amplify the gene of interest using PCR, and this is one of the most important things that we'll be learning in this session, in this chapter basically. And the fourth is insertion of this RNA into the host cell. After the ligation and everything, we will basically put this DNA into the host cell. And from the host cell, we'll be getting out into the foreign gene product. Then we will receive after this gene incorporation into the host, then we will get our product, the gene product that is like in this case, let's say it's the insulin. And then we'll do the downstream processing, which is like purification of this product, for the human uses. So now we want the DNA. Now we all know the DNA is in our cell. DNA is in the nucleus in terms of eukaryotic organism and nucleoid in the prokaryotic organism, and then which in turn is in the cell. Now, in order to retrieve this DNA, we need to break this. We have to enter this cell, and for to entering, we need to break this cell wall. Now, the bacteria, the fungus, the plant, and animals all have different cell wall. So, animal cells do not have cell wall. That is, they directly have the plasma membrane, right? They have only cell membrane, not plasma membrane. They do not have a cell wall. So they can easily burst in like abnormal friction mode. But this plant cell have a cell wall made up of cellulose, and these fungal cells have this cell wall made up of chitin, and similar with the bacteria cells. They also have a hard prokaryotic teconic based cell wall. So in order to break these cells, we need special enzymes to digest them. So there's the first part is the isolation of genetic material. We can isolate from the tissue, leaves, or the bacteria, right? And also plants or the bacteria. So in order to burst up, because this cell has a cell wall, is very hard, and after the nucleus, we'll have a lot of vacuoles, peroxisome, Golgi packets, a lot of macromolecules basically. But our focus is basically the DNA. And apart from any DNA that we are desiring, if anything comes up, it is basically so any cell they will have the DNA inside of it. So we need to break open this. We need to break open the cell wall, and then apart from all the macromolecules, RNA, DNA, protein, we just have to isolate the DNA. So this is the target that we have to isolate. There's the DNA, the rest of them. As for us, is basically contaminant. So I've already explained this to you that stating that anything that what we desire, anything apart from that will be a contaminant for us. For example, you want isolating a bacteria 1, but if bacteria 2, 3, and 4 are coming up, then that means they are contaminants for us. But if someone is working on a bacteria 2, for them bacteria 1, 3, and 4 will be contaminants. So based on whatever I am studying, everything except those things will be a contaminant for me. Now, in order to break the cell wall and then and to bring out the DNA, we can treat the bacterial cells or animal cells with enzymes of the lysine, cellulase, chitinase. So, since the plant's cell wall is made up of cellulose, it can be degraded by cellulase. And the fungal cell wall is made of chitin. It can be again broken down with the help of chitinase. Right? And once this bacterial cells or the plants or the animal cells burst out, they basically degrade, or the cell wall degrades. The cell is broken. It can release all the DNA and other micromolecules such as RNA, protein, polysaccharide, lipids, everything. And whatever we will get everything in the mixture. We'll get protein, we'll get RNA, the DNA, almost everything. Everything whatever was there in the cell. So remove all those kind of contaminants. To remove those contaminants, like what are the contaminants? As I mentioned, we have a lot of things here. We'll have the DNA, we'll have the RNA, we'll have the protein, we'll have the lipids, etc. Right? So this except DNA, whatever, because we desire the DNA, everything else will be treated as a contaminant. So remove the RNA, we can treat it with ribonuclease. Ribonuclease. So we already know that what is meant by nuclease. So this ribonuclease, but essentially it will specifically indicate the ribonuclease. RNA. Then to remove the protein, it is treated with the proteases. So with that help, we can remove the protease and then extra. And then everything will be removed with the centrifugation or using salting out method, salting in method. And then when we add a chilled ethanol, the DNA precipitates out as a fine thread in the suspension. As I can see here, there is the DNA molecule. So we have this cell. Let's say this is bacterial cell or fungal cell or plant cell, right? This will have a DNA. This will have a vacuole, Golgi, everything. So first, our first goal is to break open the cell wall. For this, for the bacteria, we'll treat it with lysozyme. For the fungus, we'll treat with chitinase. And the plant will be treating with the cellulase. So since the plant membrane is made up of cellulose, chitinase is for the fungal is made of chitin, and then we have bacteria which has its own cell wall. After this, after treatment with this, so these are the enzymes, right? Always remember these are the enzymes that are helpful in cell wall degradation. Then after this, we'll have everything in the media, like everything in the bucket, DNA, RNA, and protein. And since we desire DNA, we'll remove the RNA by treating it with ribonuclease, protein with proteinases or proteases. Anyway, after removal of this RNA and the protein, the DNA can be isolated by precipitating it. So we learned that DNA is a we learned one thing about the DNA that DNA is a hydrophilic molecule. So the second we add ethanol or propanol, it will basically this like precipitate in the tube itself. So after the precipitation, we can spell it down, or you can just take it out. So this is the data set. So this was the entire process. So in a summary, what I'm trying to say, and this is what you need to memorize because it's often asked about the process of the RDA technology is that first you need to remember the cell walls, and to break that cell wall, what kind of enzyme is used. So to break the bacterial cell wall, you have to use glycozyme, and to break plants cell wall, we need to use cellulose, and then for the fungus, we need to use chitinase. And once the cell wall is broken, all the micromolecules will come out, and then RNA will be removed by ribonucleases and protein by proteins. And as a whole, this ethanol is added, chilled ethanol is added, which will lead to the precipitation of the DNA, which will lead to the precipitation of the DNA. now, now we have a after the precipitation, now we have a clean DNA, right? I'll just put it like this. So this is our DNA, double-stranded DNA after the purification. Now, after this, we have isolated the entire genome, right? Like we after, let's say we are still focusing on the insulin, so we have the genome like this. I'll make it a single strand right now. So we have the genome here, genome, and genome. So basically DNA, like it's very, and we have our three base pairs, the DNA. So this is the DNA. Now we need to understand where is the insulin because our target is to isolate insulin. So let's suppose here the insulin is there. So what I'll do is my next goal after the isolation of the DNA is to isolate this gene itself. Isolate the gene of interest. And to do this, to isolate it, we'll again use molecular scissors. So purified DNA is incubated with the restriction enzyme. As a result, DNA is digested. So we use a restriction enzymes in order to cut our DNA or cut that thing. So those kind of things that's what we do. So after the digestion of the DNA, a technique called gel electrophoresis. Again, this is one of the important topic is gel. So in order after this cutting of this DNA, we need to separate this DNA fragment. So after this cutting of the DNA, I showed you that we have a 3 billion base pair of the DNA, but in that the specific insulin gene might be about let's say 200 base pair. So once I get this 200 base pair, we need to separate this 200 base pair from the rest of the 10 base, 1000 base pair, right? So let's say that there is this DNA strand, right, which is of which is of 2000, which is of 2000 base pair, and in this 2000 base pair, right, the 100 base pair is your DNA, insulin gene, right? So let's say this is the insulin gene, 100 base pair. So after isolation of the DNA, you cut this gene, right? So we'll get two fragments, like we'll get three fragments, sorry. Since it's cut in the middle, we'll get 100 base pair fragment, this one, and then we'll get one this fragment and one will be this. Basically, three fragments, we'll get three fragments. So let's say this is 200 base pair, 700 or so. So three fragments, right? These are gene of interest, 100 base pair, then we have 200 base pair, or let's say 300 base pair, and then 600 base pair. Now, how do I separate this from my now this my solution after the because I have in the data I added my inside. Now the solution has this, this, and this. How do I separate this? So in order to separate this, uh, so we have this 2000 base pair gene, DNA, and this 2000 base pair we have 100 base pair R gene, 300 base pair, and resources 600 base pair. Now, in order to separate, like now I know that in my solution, I'll have three fragments like this. Now I need to get only this product, only this. So for this, I'll use gel, agarose, gel, electrophoresis, right? So what it is, this is a method to separate DNA based on its size. So based on the size and charge, the DNA can be separated on the gel. So this is what we use. So in this electrophoresis, we use agarose gel. This agarose is a polymer obtained from algae. And after this, we make a gel. And using this gel, we'll like once we will load like this, something like this. We'll have wells, and then in one well I'll load my sample, and here I'll load my lab. Water is basically a concentrated form of DNA molecule which will have different sizes. So once I will I'll load my lab, I'll load my sample, I'll run it. So when I run the sample, based on the size, it will be separated. The small, always remember the small and the lightest will run fastest. Then the large or the big one, big or the large will run slowest. So let's say we have 300 base pair, then we have 300 base pair, and then we have 1.6 base pair. It's like kilo base pair. So if I run it on gel, I'll have 3 points. One will be like from here. Let's say here is 1.6. Then let's say this is so based on the size, this DNA will be separated. The smallest and the lightest will run the fastest. So in this three fragments, the smallest is 100 base pair. So this will be the 100 base pair, the fastest. And after this, there will be 300 base pair, and then I'll have a 600 base pair. So this is what the gel electrophoresis does. It will differentiate your DNA based on the size of the molecule. The smaller the size, the fastest it will run, and it will be at the towards the it will run the fastest, and then we'll have the based on the more the size, the more slower it will move. So this is the basic of electros gel electrophoresis. So now the purified DNA is included after this. Then the DNA fragment is separated by a technique called gel electrophoresis. So this is called agarose gel electrophoresis. Yeah. And this agarose is a polymer purified obtained from algae. Now this gel electrophoresis is employed to check the progression of a restriction in the branch. So we know that after digestion we'll get different enzymes, right? So after this we'll obtain a variable banks. As you can see here, as I mentioned, these are the wells. And if I load the sample, then the largest band will be behind, while the smallest will go fastest. Here is the smallest. Now, again, I told you that it will be separated on the chart as well. So now we know that DNA is negative charge. Negative. So if it is negative charge, then it should go towards positive anode. That is positive anode. So this is what happened. So when we make a gel, we put it in this kind of tank, and we'll orient in such a way that the wells are towards the negative charge, that is cathode, black, and then this will be in the positive charge, that anode, right? And since the vein is negative charge, it will easily move towards the anode. So yeah, so this is what they are trying to talk. That DNA is negative charge, so it will move towards the anode, which is a electrolyte is positive. And this data fragments are separated according to the size to sieving effect of agarose gel. So this is a sieved algae. So the separation is because they are in the agarose gel. So like, what is the principle behind the separation? So first, the movement is because of the negative charge. Like since the DNA is negative, it will move towards positive. See, here the DNA is loaded, and then it will move towards positive. And second is that this aqueous gel has pores in it, and in this pores, the DNA is able to pass uh, accordingly, and because of that, the separation is there. For example, let me tell you, like, okay, now I have made an aqueous gel. Now the concentration might depend. Like, I mean, like, can be different. For example, let's say I'll make 1%, 2%, 3%, 4%. Right. Now, the higher the percentage I go, the smaller it will be the spores. That is, when I say that it has a sieving property, sieving, I hope you know that sieving, like, we filter our tea, right? So that is sieve. So sieving basically means having a presence of a holes, and based on that, there is separation. So it has pores. So if it is 1%, let's say this pore, if it 2%, then again this gets smaller, smaller, smaller. Let's say 10%. So in the pore size, right? So if I if I made a percent of 1% gel, so the smaller molecules, let's say this is 100, this is 500, this 800, 1 kb, 1000 base per. So they will pass. So if they pass, the smaller one will be the smaller one will pass easily, and there will be no resistance. So they will move really fast. And after that, there will be 500 base per ladder, side, and there will be a bigger one, a little bigger one, and a little bigger. I'm just depicting you the size. Actually, in gel, it will not all the same, like original we had said. Similar to the next slide I'll show you. So based on this sieving property, based on this pores in the gel, the DNA basically passes slowly. So that is the property of the agrosine. What is the property of agrosine? Agrosine is obtained from seaweed, that is algae, gelatin and gelatin and something like that. I forgot that exact name. So algae, and it has the property of sieve property. That is basically termed as pores. And therefore the DNA will easily pass through these pores to reach the uh, the other side. And this is the one property of this. Second, that we know that DNA is negative, which are most of positive, and that is anode. So this is how the electrophorese works. So after the digestion of the DNA, it is loaded on the gel, and then it is separated based on the size, and we take out the desired band. The same thing is done with the vector DNA as well. So yeah, so there's an example. So there's a DNA sample, then I added recipe there, and it has cleaved this DNA. So this was the DNA, it has cleaved into 3 different parts, and then I ran it up then I'll get this kind of 3 lines. So here, this is a depiction of gel electrophoresis. First lane, there is one. It is lane one. There is no digestion. This is without enzyme, basically without enzyme. The second set two is a digested. 2 to 4 is a digestive, and as you can see, how the smaller bands really move fast, but the bigger ones stay behind. Now, how do I visualize this? So you can see this bike. This is very prominent bike. So how do you visualize this? So this DNA fragment can be seen as a bright orange. So we already know DNA is like such a packed molecule, such small molecule that we cannot see it by with a naked eye. But we can visualize it. And with this agile electrophoresis, we have another thing coming up, that's ethidium bromide, which happens to bind to DNA and illuminate in presence of UV. So DNA fragments can be seen as a bright orange colored bands when they are staying with ethidium bromide and are exposed to UV radiation. So what happens here is that the DNA, this let's say DNA, and there's this one more compound called ethidium bromide. And what it does is basically it will go and bind with the DNA. And this has an excitation emission spectrum. So once it binds to this DNA, when we give a UV light, it will radiate a bright orangish color. And this is how we were able to visualize the DNA. And for example, if this is my desired band, I cut this band, I melt the gel again, isolate this fragment. So that is how the electrophoresis was happening. So let me just summarize this, and then we'll end up today's session. So we started with the process of RDA technology. For example, to isolate a gene of insulin, because I want to make insulin at a large scale, for which I first isolated by DNA. And if the DNA is isolated from bacteria, we have to use lyozyme. If we isolate the DNA from fungus, we need to use chitinase. And if we use from plant, we need to use cellulase. And after the isolation of the DNA, after this like breaking of the cell wall, all the macromolecules will come out. There is RNA, DNA, protein, etc. Macromolecules will come out in the come out. And once this macromolecules come out, we can remove this macromolecules, such as RNA, by treating with ribonuclease, and with the protein that will come with proteinase. And once this is out, once this is degraded, we can precipitate DNA with gel ethanol. Like after this, like we have already isolated our DNA, test. Now we know that the DNA is very big, and in this big DNA, the genome will be very small. For that, we'll basically use molecular scissors, that is molecular scissors, and cut our DNA. After cutting of the DNA with restriction enzymes, we will after cutting of this or cutting of DNA with restriction on bounds, will separate the desired band on a gross gel. And the technique is called gel electrophoresis. So we are using electric field and the gel in order to separate the DNA. So what happens here is that we have cellulose obtained from seaweed, this algae, and based on sites and charts, we separate the DNA. So this is an agrosel, and I hope you remember the concept of the percentage. The higher the percentage of the agrosel, for example, 1% is 100 ml, 2% is 2 gram and 100 ml, 3 gram and 100 ml, 4 gram. So the higher the percentage, the smaller will be the size of the jet, and the better will be separation. So this I made 1% gel, and I loaded my DNA after digestion, and I have a pre-digestion. Like my expected size is 2 kV, 100 base pair, 50 base pair. So based on the size, we know that the 50 base pair is the most small, it will go up. Then we'll have a 100 base pair, and then we'll have a 2 kV, we get 2000 base pair, 2000 base pair, 100 base pair, and 50 base pair. And this is how it will run. And we know that DNA is negatively charged, so it will move towards the anode. So that's it for today. We'll meet in the next session, and I'll be continuing with this lecture series again. In the next session, we'll be continuing about this process behind the RNA technology. And yeah, so thank you for joining today. Let's meet in the next session.